So the nervous system PowerPoint is a pretty lengthy PowerPoint, so I'm going to divide it up to two so the videos aren't too long when I go to download or upload them up. And so this will be part one, but basically we're going to be looking at the nervous system, we'll be looking at the tissue, and basically how the neuron operates through signaling. Later on, we're going to get into the different parts of the nervous system, like the sensory and the autonomic nervous system, but this will be specifically on the generalities of the nervous system and the operation of the classic cell from the nervous system, the neuron. So since we have 36 trillion cells that are having to operate together in our body, we have to have means of communication, sometimes communication over long distances. We also need a system to kind of organize and control the functioning of these cells. And so the nervous system, and later on we'll talk about the endocrine systems, are those two systems that are going to be involved in communication and allowing coordination and control of the tissues or the cells in the body so they can all function as one or function together. So we'll first look at the breakdown of the nervous system, and then we'll look at the tissues or the cells of the nervous system, and then we'll get into the operating of the neuron itself. But looking at the nervous system, we got two big categories or two classifications. That's the central nervous system, all right, and that will include the brain and the spinal cord here. Okay, the rest is the peripheral, and that's going to contain the nerves, okay, which are just basically bundles of axons of the neuron and cell bodies, but everything outside of the brain and the spinal cord is going to fall under the peripheral nervous system. And that can be divided into two categories. We got the sensory or the afferent, and the function of this is to bring information from the outside, the peripheral, and bring it towards or into the brain or spinal cord. So in this direction. So the afferent. Sensory neurons would be afferents because they're bringing information back to the brain. Okay, the efferent neurons are going to fall under what we also call motor. Okay, so that's going to bring information from the central nervous system out to the periphery and going to talk to the various organs, going to have control over the different tissues throughout the body. Okay, so sensory allows us to detect, bring information in. The brain's going to allow us to integrate it, and then the brain can send out messages through the efferent to help control the various organs through the body. The efferent neurons, nervous systems, can be further broken down into the autonomic and the somatic motor neurons. Okay, the autonomic neurons, these are the ones that operate. This is also called, autonomic is called the involuntary, because these are things we don't have to think about. These are our basic functioning items that are going on without our actual conscious knowledge. Like, I don't have to sit here and say, heart beat, beat heart, beat heart. No, I don't have to tell my heart to beat. I don't have to consciously think of that because that is under control of the autonomic nervous system. Okay, the autonomic can be further broken down into the sympathetic and parasympathetic, and we're going to spend a whole lecture on this autonomic nervous system and looking at these two branches, the sympathetic and parasympathetic. Okay, but what tissues are under control of the autonomic? Cardiac muscle, right? I don't have to tell my heart to smooth muscle, a lot of which you'll find in your digestive tract. I don't have to tell my digestive tract to move food through. It does it automatically. Exocrine glands, I don't have to tell my body to sweat. Some of the endocrine glands are also under control, and also adipose tissue. So many of those tissues we don't have to think about controlling are controlled or fall under this autonomic nervous system. Okay, the somatic would be called voluntary. That's going to control our skeletal muscles. When I want to contract my biceps and do a curl, I have to think. I have to voluntarily control it with my brain to cause my muscle to contract. Okay, we'll spend a little time on this, but we're going to spend a grand time on autonomic nervous system, and we also will spend a lecture on looking at sensory, okay, the afferent system. So this slide just gives you a written kind of basically what we just talked about on the previous slides, okay, giving you an idea of the different breakdowns of the systems and what they control of. Okay, so now let's look at the actual tissue, okay, what makes up the nervous system. And there's going to be two types of cells. The one you guys commonly probably know most of, seen in anatomy, the neurons. Okay, that's these guys here with the long, okay, these branching dendrites, cell body with this long axon. The others are the glia, although we don't think of these as much, or they're not heard as much because we always seem to focus on the neuron. The glia are basically the cells in the nervous system, and most of their functions is to make sure that these neurons can operate. Okay. They are more abundant. There is a lot more glia cells than there are neurons in the nervous system. Okay, but basically their function, most of their functions lie in operation or helping to operate the neurons. Okay, so the neurons are going to be the ones that are involved in the signaling, where the glia cells are basically the helper cells of the neurons. Let's first look at the neuron. Okay, we got the neuron, basic parts of it. We have an input, so the neuron's going to receive information and then it can integrate and do an output, signal out. Okay, the input in, where information's coming in to the neuron, is we have these branchings. Okay, these spines here, dendritic spines. These branches make up the dendrites. Okay, neurons, you can see down here, can come talk to, send information and signal to the dendrites. Okay, this neuron would be an output signaling to the input in of this neuron. Okay, input can also come in at the cell body. All right, and then we're also going to get integration. Chiefly the integration is going to take place at this region called the axon hillock. Okay, so we got the dendrites, we got the cell body or soma receiving in, and we'll have integration taking place, and we will talk about this location, the axon hillock. This is basically also known as the trigger zone. This is where we're going to trigger the output signal, and the output signal is going to be an electrical signal called an action potential. Okay. And so this is the long axon, this is the output, sending down, and then we can signal or talk to another neuron in the output signal. Okay, along the line we'll see these myelin sheaths that may be wrapping the axon. Okay, but the axon comes down and will divide out to axon terminals. Okay, and this is where there is not a junction, but they come in close proximity with the synaptic cleft. There is a small space in between this neuron terminal and the dendrite or cell body of this other neuron. Okay, but this is where the chemical signaling is going to take place, and we'll talk about that later in the lecture. So the cell body is kind of the control center. That's where we're going to get integration. right? And this houses all those organelles that the common cells have, right? And it has the nucleus to making the proteins, right? So it's receiving, it's got all the organelles, and it also will do integration. Chiefly the integration again is going to happen where basically the axon and the cell body meet up at the axon HELOC. So here's the dendrites. Okay. Again, we've already discussed this is going to be the receiving or input end of the neurons. Okay. Gives it lots more space and surface area to receive information. So receive that incoming information from neighboring cells. Okay. And then the output end, that's going to be the axons. Okay, the axon is where the electrical signal or the signaling is going to take place. And these can be very long. They can be up to several feet long. And so it allows the neurons to signal over long distances. So this is pretty unique about the neurons. They can be microscopic in shape, but they can be several feet long. And usually the length is in these long axons here in which the electrical signal is going to take place. Remember the initial stretch of the axon, right, where the cell body and the axon meet up is the axon HELOC, right? And this is going to be the trigger zone. This is going to be kind of like a switch. It's going to be a switch whether this electrical signal, the action potential, gets sent or not. Okay, so this is going to determine whether this neuron is going to communicate with whatever tissue it is talking to. Okay, we'll talk about voltage-gated sodium channels. They are in high concentration here. Just kind of reminding you, channel. So what's going to go through there is sodium. Okay, but it is gated, meaning that it is not always open. Okay, so if it's closed, sodium cannot move in. But voltage, we will see there is a change in what we call membrane potential. Usually the cell is at negative 70 millivolts. It is more negative inside. But if this changes to a more positive number, a more positive voltage, that can cue these gates to open. So we'll get into great lengths and details of these channels, but just remember this is kind of the initial stretch where we see those sodium channels. Okay, so the axons, they're going to come down. The action potential can be sent down, and then we reach the end of the axons here where they branch. These are the axon terminals. Okay. There is sometimes branching up near the axon, up further up, where a new axon comes off, forming a collateral, and then that collateral would come in and branch off to terminals. Okay, but this is less common. Usually it is one axon coming, and then the branching doesn't take place until we get to the axon terminals. Okay. This is where our neurotransmitters, okay, the chemicals can get released to signal. Okay, we can get what we call chemical signaling. Okay. So the neuron can signal to whatever tissue it's talking to, okay, through these what we call synapse. Okay, so the neuron does not, these terminals do not touch the tissue. They come very, very close, okay, but they form a synaptic cleft. Okay, and later on, like I said, we will get into the signaling. So the last part of this nervous system lecture will be the signaling that's taking place here at the synapse. So in the peripheral nervous system, those axons make up the nerves that are out there. Okay, so they are just basically big bundles of the axons. Okay, so they are pathways of the signaling. Okay, carries information either towards the brain or away from the brain, or the nerves can be a bundle of both. And those are termed sensory. Okay, that's going to carry information from the outside, from the sensors in the body, to the brain. Those are the afferent signals, where the motor neurons or nerves are going to carry efferent signals, so basically the motor signals out to the various organs, either skeletal muscles, hearts, etc. Or the nerves can also be a bundle of carrying information in both directions. Some neurons may be carrying information in, others may be carrying information out, the efferent pathway. Okay, but again, they are just bundles of axons of the neurons. So classifying these neurons, we can go functionally like the sensor. Okay, the sensory is the afferent, just as we saw on the previous slide. Okay, and basically the pathway is sensory information is going to carry information in the afferent neurons. Okay, it's going to be sensory information like temperature, pressure, light, so forth. Many different senses that can be carried through these afferent neurons. And then inside the central nervous system, we have a class of neurons called interneurons. These lie entirely in the CNS. These are basically communicating or integrating neurons. They allow integration between the sensory signal and the efferent, the motor neurons. Okay, so that's the basic flow. We get sensory information coming in, the brain or spinal cord can integrate it, okay, and decide if we need to make a motor response, whether we need to move away from something, use our skeletal muscles, we need to raise our heart rate. Just depending on what sensory information it is, brain can integrate that and then send out the appropriate response through the efferent neurons. Okay. So the interrelation, we can have the sensory, just kind of the flow, sensory within the central nervous system, and then we can make a response through the motor pathway. The sensory information can come from various sources. Okay, it can be visceral, okay, blood pressure. Our baroreceptors record our blood pressure. Special senses, those are the ones that are, and we're going to spend a whole lecture going over special senses. Those are the ones make up our eyes. Those are the ones located in our head region. All right, and then the somatosensory are the general senses throughout the body, like our cutaneous. Throughout our skin, we have sensory information. Okay, but this can come of various types depending on the neuron, depends on the sense and what sensory information it is bringing into the central nervous system. Okay, the integration, we have those interneurons in here in the brain and the central nervous system, and depending on the sensory information, we can send out, the brain can send out a response. We can go through the somatomotor if we need to move away or we need to move our muscles. The brain would send a response through the efferent somatic motor neurons. Okay, if it's something like heart, we're trying to control blood pressure or so forth. Those are things we don't have to voluntarily think about, and those are controlled by the autonomic motor neurons. Okay, and we're going to spend a lecture on this pathway here, the autonomic, but it is a motor pathway sending out to basically smooth muscle, cardiac muscles, and glands for the most part, and also adipose tissue. Right, besides the neuron, there also lie the glia cells throughout the body. Okay. The glia cells are basically the support cells or helper cells. Basically, most of them are there to support the neurons. The neurons are the big signaling cells throughout the nervous system, but the glia cells are there to support and help maintain the functioning of the neurons. Okay, there are much more. but looking at the different glia cells throughout the body. Okay. We have the ependymal cells. These are the ones that basically make they're the barriers between compartments, and also there are specialized cells that make that cerebrospinal fluid, which you probably learned about in anatomy. The astrocytes are kind of one of the big players of the glia cells. They do a lot for the neuron. They're kind of like the personal assistants of the neurons, maintaining the environment. They also help glue the neurons close to the blood supply. They also are functioning of making the blood-brain barrier. Okay. And then there are also the microglia. These are the resident immune cells in the brain. Okay. Again, they also scavengers. They're gonna, if anything foreign comes up into the central nervous system, the microglia are there to start the immune response. And lastly, there are the oligodendrocytes. Okay, oligodendrocytes are going to make what we call the myelin sheath around the axon region. Okay, and we're going to talk about this when we get into our action potential, the functioning of this. This actually allows the neuron to make a much quicker signal, is because of this myelin sheath that basically makes a fatty insulation around the axon. Okay, so these four are a part of the central nervous system, the CNS. Okay, there are two other out in the peripheral nervous system, so out in those nerves, where those nerves lie, so forth. We have the support cells, kind of like the astrocytes of the central nervous system. The satellite cells are kind of like the personal assistants out in the peripheral nervous system. Okay, and support the cell bodies of those neurons that are the cell bodies that are out in the periphery, and they also give kind of like a padding. Okay, they will surround, these cells will surround the cell body of the neurons out in the peripheral, because you realize that the periphery, the neurons don't have any bone around them. The central nervous system has a skull, has a vertebral canal, that they have these bony structures that surround and help protect them. Whereas in the peripheral nervous system, the neurons don't have that protection, and so satellites add a little bit about that by surrounding the cell bodies. Okay. And then, similar to the oligodendrocytes, the Schwann cells out in the peripheral nervous systems are the ones responsible for the myelin sheath around the neurons. Okay, so basically the same functions, oligodendrocytes and Schwann cells for the most part, making that myelin sheath, just two different cells in the two different parts of the nervous system. We're not going to spend great lengths in the glial cells in the physiology class, but two we're going to be seeing a bit about are the astrocytes. Okay, because the astrocytes, you can see from all their functions here. Okay. They can take up, release chemicals in the synapse. They're going to provide the neurons with ATP. Your neurons are very highly use up ATP, and so they must have the substrate for that production. Maintain homeostasis, so they maintain that environment. We're going to see when we look at the neuron that that environment has to stay very homeostatic for the neuron to be able to perform its functions, performing those electrical signals we'll be talking about. Okay, one of the other, talk a little more in detail, the blood-brain barrier. This is going to regulate, so not everything that gets into the regular bloodstream is allowed to have access into the brain, because there is this blood-brain barrier, and the astrocytes help form that, and we'll see that in a second. Okay, growth factors, and they are a sense of neurostem cells. So you can see this is multifaceted. It is kind of like they are the personal assistants of the neurons. They're there to basically make that neuron function and a lot of the central nervous system in functioning. So here's a closer look at the blood-brain barrier. We already said the astrocytes were part of this, and basically it's made up of, when we get to the blood vessels that supply the central nervous system, we'll see these are gray are the epithelial or endothelial. Those are the epithelial cells that line the blood vessels. You see sometimes these blood vessels are kind of porous. There is space in between the cells in which materials can get through. In this case, we have these tight junctions here, so material cannot just flow in between the cells out of the blood vessel. Okay. In this case, for the barrier, whatever needs to come into the environment here in the central nervous system, whatever has to come through must first come through the endothelial cells, and then there is a second mode of protection in these foot processes of the pink here is the astrocytes wrap around here to also further exclude things. Okay, so not everything that is out here in the bloodstream is going to make its way into the central nervous system, okay, due to this blood-brain barrier. The other neuroglia cell we're going to see later on in the lecture is going to be not so much the oligodendrocytes, but we will see the Schwann cells fairly regularly. Okay, because we're going to be talking about myelin. And myelin is just basically like an insulation. You know, your electrical wires, they have that plastic or rubber wrapped around it to provide insulation around so you don't get zapped by the wire. Remember, these axons are sending electrical signals, and this insulation is going to help keep that signal inside the axon. Okay? And what they do is they basically wrap around and squeeze out the cytoplasm, so what's left is basically this almost fatty layer, this myelin sheath. As you can see here, the myelin. So this is just a bunch, okay, phospholipid membrane. It is a fatty layer. So they wrap around, squeeze out the cytoplasm, and so what's left is this just wrapping, this multi-layers of phospholipid. Okay. And so you can see the Schwann cell nucleus here. Here's what's left of the cytoplasm, but the rest of this is just wrapping of almost like electrical tape around an electrical wire. This is just wrapping of the plasma membrane around the axon. And when we look at the axon, each of these are an individual Schwann cell here, but they are not wrapped. There are spaces in between here that have exposed axon. Okay, these are the nodes of Ranvier. Okay, so we have insulated the axon here, but there is still open regions in the nodes of Ranvier in which there is open axon. Okay, we are going to look at how this affects the signaling of the axon. Okay, it is actually going to allow the neuron to make a lot quicker signal. Okay, it's going to allow the signal to actually jump. But we'll get in that when we start talking about the action potential. We'll look at the electrical signal down this axon, and then we'll look at this electrical signal down a myelinated axon and see how we get this more rapid signal. So let's start getting into the electrical properties of the neuron. So remember that action potential. Basically, this neuron is going to signal. One of the first signaling is an electrical signal. We will get, when we get to the synapse where it talks to the other cell, we'll talk about chemical signaling, but throughout sending signal down the axon, it is an electrical signal. So we have to get into some physics or electrical properties of these cells. Okay. So nerve, muscles, two of the cells that are excitable that can actually transmit electrical signals. So excitable, let's see, cardiac muscle is also excitable, skeletal muscle, all the muscles are excitable, and the nerves are also excitable, and they're able to basically propagate, send electrical signals rapidly through the cell. Okay. So let's get into the property of the neuron and actually any cell in the body. If we were to measure, say we had this bath outside the cell, okay, say this was made to look like the interstitial fluid, and we had the cell. Okay, if we were to actually take a voltmeter, put it in the solution, and put it inside the cell, what we are going to find is that in comparison to the reference electrode, the cell is more negative. All right, roughly around negative 70 millivolts. Okay, so between the inside and the outside, it is more negative in the cell. And kind of just looking at what took place in that previous slide, if we had a reference electrode and we have it out in the solution, we're not having any change in voltage. But then once we push the reference electrode, okay, or sorry, the recording electrode inside the cell, you can see we get this drop, roughly about negative 70 millivolts. Okay? And this correlates with what we call the resting membrane potential. Okay? Because when the cell is at rest, the difference in potential, okay, voltage between the outside and the inside is around negative 70 millivolts. And what is actually being set up here, we're going to talk about, is what we call an electrochemical gradient. We've seen chemical gradients, right? Concentration gradients with chemicals, but we also have a gradient in the electrical property, the difference in voltage between the inside and the outside. Okay? You've seen a chemical gradient in that we're going to start using this. You guys have seen this before. We have sodium high outside and sodium low inside. Okay, so that is the chemical gradient, but we also have an electrical gradient. It is more negative inside than it is outside. So what is going to be, basically, why is it? Why do we see this negative charge inside the cell? Why is it more negative in here? Well, one of the things we've already seen is there is uneven distribution of ions. Our sodium is high outside and low inside, and our potassium is high inside and low outside. Okay, so we have these distribution of ions help to maintain this negative environment in the cell. So, two factors. We've already seen the difference in ion concentration. Okay, is going to be the first part of what's maintaining this membrane potential. Okay, other factors are you have these large negative fixed ions in here. Okay, but the real main factor in keeping this membrane potential in that negative 70 millivolts range is the difference in permeability. Okay, remember these ions are not allowed to, potassium can't just zip out of the membrane. Okay, so there are channels, open channels at rest that allow ions through. There are sodium channels, or multiple channels, but what there is there are much more open potassium channels than there are sodium channels. So if there are more channels for potassium, which compared to sodium, which one's allowed to move more rapidly? Yeah, there's going to be more what we call efflux out of the cell of potassium than there is influx of sodium into the cell. Okay, so there is a lot more positive ions leaving the cell than there is positive ions coming in. So this permeability to potassium, the greater permeability to potassium at rest. Again, this is at rest, because at rest these open channels are open. Okay, because we're going to be bringing in some what we call voltage gated channels into play. Okay, but this negative 70 millivolts, we have uneven distribution of ions. This causes concentration gradient, and there are channels for potassium and sodium that are open at rest. But again, there are much more potassium channels. Therefore, the cell is more permeable to allowing potassium out than it is to allow sodium in. Okay, and that is the one really main factor in drawing the membrane potential down to the negative 70 millivolts. So now that we see that rest, the membrane potential is roughly around that negative 70 millivolts. What we're going to see is there are changes to this. There are changes that can take place that can actually bring the membrane potential either more positive or more negative or even greatly negative. And that's going to be, we have depolarization. You can see depolarization is taking place here. The membrane potential, the difference between the outside and the inside of the cell, the inside of the cell is becoming closer to the outside, becoming more positive, closer to zero. Okay, and so depolarization, see, taking place in a positive direction. The other term, repolarization, is after depolarization, the return back to resting membrane potential. Okay, going now back into the negative direction. We went positive, now we're coming back in the negative direction. That is the repolarization. And there are times where the membrane potential actually can be hyperpolarized, and that would be in a more negative direction than the actual resting membrane potential. Okay, so hyperpolarization is below resting membrane potential. Depolarization is in above or a more positive direction from resting, and repolarization is returning back to resting membrane potential after depolarization. Okay. So we got several things at play. What we see is we have an electrical gradient. We have a difference in electrical properties from the outside to the inside. So we have an electrical gradient, and we can do this because we have the cell membrane. It allows us to separation of charge, okay? And it allows us to create this electrical gradient. Okay, as we see, it's more negative inside than it is outside. There is a gradient, an electrical gradient. Also, the membrane allows us to have a concentration gradient. We've seen that the ions are more sodium outside than inside, and the opposite for potassium, more potassium inside than outside. So this separation of charge and of ions allows us to maintain these gradients. And overall, it allows us to have what we call an electrochemical gradient. Electric, and a chemical, the concentration gradient of the ions. And so what's giving us that electrical disequilibrium? As we saw, we have that chemical disequilibrium, right? And we've seen the permeability also, but the electrical net charge between the two regions. And one of the main factors is the uneven distribution of the ions. Remember, our sodium's high outside, low inside, potassium the opposite. So that helps give us that electrical disequilibrium. And we can see that chemical disequilibrium is helping maintain this. You can see here, I is for interstitial in this case. C is for what's in the cell. Okay, and you see sodium is high outside in the interstitial fluid compared to inside. And potassium is the opposite. Interstitial fluid is very low, but it is high inside. Also, we'll see bicarbonate, but another thing we saw were those large anions, those large negative ions. You can see there are much more inside the cell, so those also help create that electrical gradient that we are seeing in the cell. Okay, the ones we're going to be specifically talking about when we start talking nervous system, we're going to be focusing mainly on those sodium and potassium. Okay. So why do we care to know about an electrical chemical gradient? We do because these are the two forces at play that are going to dictate the movement of ions in or out of the cell, okay, for diffusion. And we have a chemical gradient. Remember, in concentration, it's going to move from, the ion wants to move from high to low concentration. There is a force there driving the ion in that direction. But we need to add in the electrical gradient. Remember, positive repels positive. If you have two positive sides of a magnet, they repel each other. But negative attracts. So there are forces here at play. And if you remember, what are our ions? The ones we're going to be chiefly working with are sodium and potassium. Okay, they are both positive ions. But a negative environment will attract positive ions, and a positive environment will repel positive. Okay, so keep that in mind as we're looking at the forces that drive whether the ions are going, the movement of the ions in or out of the cell. So Mr. Nurse, since this is what we call the Nurse equation, is able to determine at what we call our electrochemical equilibrium. All right, and that is telling you where, okay, this is, say, the force for my concentration gradient, maybe drawing my ion, say sodium, in this direction, where the electrical force may be pushing in this direction. Okay, and the electrical force is determined by what the membrane potential is. Okay, that's going to be the electrical force. Is the electrical force negative? Is it positive? What's the membrane potential? Okay, so what the electrochemical equilibrium is telling you is when the force that is drawing, the chemical force, the concentration gradient that's drawing sodium in one direction, when the electrical force is equal to that force of the concentration gradient, when those two forces are equal, sodium will stop moving in or out of the cell, or will be no net movement in or out of the cell. Okay, and so what the Nurse equation is telling you is at what membrane potential, what millivoltage of membrane potential will these two forces be equal? Okay, because your body keeps a certain ion concentration inside and outside the cell. So when we change electrical force, at some point, the electrical force is going to be the same as the force that is drawing the ion down its concentration gradient. Okay, and so what this is calculating is where will the ion move? At what membrane potential will the ion stop moving? Okay, inside or out, depending on the ion. Okay, and we calculate this for individual ions. Okay, so we're looking here. We got electrical potential. Okay, so the equilibrium potential, the Nernst equation, is predicting at what value of membrane potential do these two forces, in this case, potassium. The concentration gradient is drawing potassium in this direction. But remember, the membrane potential is -70. If I am permeable, and then what the Nernst equation is assuming is that I'm just assuming that the membrane is only permeable to that specific ion, to potassium, or if we're calculating the equilibrium potential for sodium, it would be, we're assuming that the membrane is only permeable to sodium. So in this case, say we're trying to calculate the equilibrium potential for potassium. Assume it's only permeable to potassium. There's only channels for potassium. Okay? The concentration gradient draws potassium out because potassium is high inside, low outside. But as potassium leaves, what's going to happen to the membrane potential? It takes with it its positive charge. So we were at -70, okay, millivolts at rest. What happens when potassium keeps leaving? Yeah, the value is going to become more negative because we're losing more positive charge inside. And if you remember, negative attracts positive. So we have this positive ion leaving. As this area becomes more negative, there's going to be an electrical force. That negative electrical force is going to want to keep potassium from leaving because the negative attracts the positive. So as this number becomes more negative inside, there's going to be an electrical force that's drawing potassium this direction. And so what the equilibrium potential is saying at what number inside here, what membrane potential do these forces equal out? Okay. And you can see with the Nernst equation, we have to take into effect the ion concentrations. But these is what the body keeps. We maintain these concentrations in the body. So we can use those concentrations, those physiological concentrations, to figure out what our equilibrium potential is for a certain ion. Okay, when using the Nernst equation, when we factor in potassium, we actually find that potassium would, if we're only permeable to potassium, potassium would leave the cell until that membrane potential got to -90. Now at -90, that electrical force is trying to keep potassium in, as the chemical force is trying to put potassium out. Okay, so for equilibrium potential for potassium, it would come out to 90 if we're using the values that we see around our cells in the body. So now when we look at the equilibrium potential for sodium, remember sodium is high outside and low inside. Okay, we can see it is 61, or positive, we'll use 60, generality. All right, so this means that sodium, if the membrane was only permeable to sodium, and with the physiological concentrations, remember sodium's high outside and low inside, that sodium would rush in due to the concentration gradient, but as sodium rushes in, it's bringing a positive charge. And as a positive charge at negative 70 millivolts starts coming up goes into a positive range. And remember, positive charge repels positive. So as the membrane starts getting more positive, there is an electrical force that is pushing against sodium from coming in. All right, when that concentration gradient force equals the force of this, sodium will start rushing in. So sodium doesn't come in all the way to equilibrium. It is repelled by this electrical force. Okay, so it would only come into about, once we reach positive 60 millivolts, the sodium would stop rushing in because of its equilibrium potential, which is positive 60. Okay, you can see for these other ions, okay, the chloride is high outside, low inside, and same with calcium. Okay, we're going to start seeing these. We'll see calcium later on. Right, when we start talking about the chemical signaling we find down at the synapse, okay, calcium becomes a big player in that. Right, but for our cases, for our action potential, okay, we got negative 90 and positive 60 for our sodium and our potassium. Kind of goes back to our resting membrane potential, in that, where is our resting membrane potential? It sits around negative 70, right? And what is that closer to? You can see it is closer to the equilibrium potential of potassium, which was negative 90. Right? So we can tell that the permeability to potassium at rest, because it sits closer to equilibrium potential of potassium, that it is more permeable to calcium. There are much more what we call leak channels. Leak channels are open channels. Calcium can leak out. Okay, it's not this big rush, but basically we're leaky to potassium. We are more leaky to potassium at rest than we are to sodium. Although some sodium comes in, you remember our potassium, our equilibrium potential for potassium is negative 90. It's all right, where membrane potential sits at negative 70, not negative 90, because there are still some potassium leaking in. But again, we are more leaky to potassium. Okay, you can think about it as amount of channels. If for every 40 potassium channels I have 1 sodium leak channel. Therefore, I can leak out much more potassium because I have more channels for leaking rather than the sodium. Okay, so at rest, we are more permeable to potassium. Okay, and so that's kind of the rundown on what gives us our negative 70 millivolts. Our fixed anions, unequal permeabilities, more to potassium rather than sodium, and therefore we sit closer to that negative 70 millivolts. But there would be no leaking of ions if there were no concentration gradients. So we have to pull in the effect of the sodium-potassium pump. Okay, because Although we have channels were permeable to sodium, if there were no gradient, if potassium was the same inside and outside, there would be no leaking. But we have the sodium-potassium pump pulling out 3 sodiums and bringing in 2 potassium. And as you can see, this isn't an equal exchange. So if I'm pulling out 3 positives ions and there are only 2 coming in, you can tell this should make for a more negative environment. Okay, so all these factors are pulling together to make the negative 70 millivolts. So before we start moving on into how we are going to change that resting membrane potential, how we're going to get that depolarization or that repolarization or hyperpolarization, I want to review some of the channels. If you remember, we had non-gated. Those would be channels that are always open. Those leaky potassium channels are always open, so they are non-gated channels because they allow potassium to leak out. They are not closed. Okay, they are always open. There's no closing them. Where gated channels, gated ion channels, are only open in response to some kind of stimulus. Okay, and we can have chemically gated. That's if a chemical binds to the ion channel will be open. So you can see in a gated channel, we have a closed state. This is where no ions can go through. They are turned back. Where in the open state, they can go through. But to get from here to here, there has to be something to signal this. There has to be a stimulus. Okay, and chemically gated, it is chemicals. In voltage gated, the ones we are going to be discussing here for the action potential, it is a change in membrane potential. Okay, when that negative 70 millivolt becomes more positive, in most cases, it is going to be the cue to go from closed to open. Right? I'm going to just throw the number out here now because negative 55 millivolts. Okay. It'll be a cue to open up many of these voltage-gated channels. My membrane potential moves from negative 70 to negative 55 millivolts. That would cue this gated, voltage-gated channel to open. So remember, these neurons, they're going to have to communicate over these long distances, and to do this, they have to send signals over long distances. And so to accomplish this, they're going to be using electrical signals. Electrical signals are very rapid. It's basically a flow of charged molecules, and so we can do this very rapidly, and so that's how the neurons are going to send the signals down those long axons, is through electrical signaling. Okay. And there are neurons, you can see, and then later on we're going to see the muscles have the ability to be excitable. In a sense, they are able to send electrical signals, propagate electrical signals rapidly over long distances. Okay. And they are going to do the signaling by changing permeability to certain ions, using those channels, using those gated channels to change the permeability to various ions. And in our case, for the action potential for the neuron, we're going to be talking about sodium and potassium gated channels. Okay. And how is this done? So we got the flow of electric charge, and in this case, the charge is coming from the flow of the ions. Ions are positively charged. We're flowing electrical charge by changing the permeability, changing the ion movement. Just remember your electrical chemical gradients. That's going to tell you which way the ion's going to move. We saw for sodium, the ion movement is inside the cell, and for potassium, the ion movement is outside the cell. So to get our electrical signals, so essentially what we're going to be doing is changing membrane potential. By changing this flow of ions, we're going to be changing the membrane potential. Okay, and there's two ways we do this in the neuron. There are going to be what we call graded potentials, right, and action potentials. Kind of the main difference is where these occur. The graded potentials are going to take place in the dendrites and the cell bodies. And what we see with graded potentials is essentially they are graded. Okay? We can have the potentials, what we mean is we can get depolarization, you saw, and we can get hyperpolarization. So depolarization, repolarization, and we can also get hyperpolarization. Okay? What we see with graded potentials is the stimulus of the change. We can have a small one like this, right? Or for graded, we can have this very large one. Okay, so for graded potentials, we can have different amplitude size potentials. Right? This is a smaller depolarization compared to this depolarization here. Where when we see action potentials, okay, they're going to be of the same size. Okay, so graded, they can have various size of amplitudes, and what we're also going to see is that this graded potential can decrease as it travels through the cell, and it travels through just short distances because these are taking place at the dendrites and the cell bodies. Okay, but graded, various size, they decrease as they travel through the cell, but they're only going over short distances. Where the action potential, the amplitude is the same. But they are going over long distances. This is what's going to travel. Action potential, this is what's going to travel down the axon. That's the electrical signal. We'll be sending down those. Some of those axons can be several feet long, so we're having these action potentials take place over long periods. All right, or long distances, sorry. What you see though, if we measured the amplitude of the action potential all the way down the axon, it would be the same size. So you can see that as we move down the axon, say this was moving down the axon, we were measuring the action potential. What we see is this one that isn't smaller. It doesn't lose size as it moves through. Okay, so that's the difference from them. But what we're going to first look at is the graded potentials, how these take place, okay, what causes these, and ultimately it's going to be the graded potentials that lead to whether we see an action potential down the axon or not. So when we look at graded potentials, okay, again, they're going to take place at the dendrites, the cell bodies, okay, and they vary in strength. They can be large, or they can be small, or they can be somewhere in between. Okay, they are just variable in strength. But you can see they're only traveling over short distances. They're not traveling down these very long axons. They're just traveling across the dendrites or the cell bodies. But also, look here. What is happening over here is we said that graded potentials, okay, graded potentials decrease over time or over the distance that they travel. Okay, here's the original stimulus. You can see we have a graded potential up that brought us up from -70, more positive, to a -40. But as it travels, it loses steam, and as you can see when we get here to this region of the neuron, the trigger zone or the axon HELOC, we have a much smaller potential. Okay, it is basically around -60. So we lost from here to here. You can see how much of the signal, the electrical signal, that was lost. Okay, and what we see, graded potentials, can come of one of two varieties. We can have excitatory. Excitatory means it depolarizes the membrane potential. Okay, but the signals sent here can also be inhibitory, hyperpolarizing, or bringing the membrane potential down. Okay, so excitatory is depolarization. Okay. Inhibitory is repolarization or hyperpolarization of the membrane potential. Okay, so seen here, so we could also have one that is in this direction if it was an inhibitory signal. And this comes into play because what these graded potentials function here is to determine whether an action potential is going to take place or not. And where that gets determined is at the trigger zone. Okay, because we have to reach threshold, negative 55 millivolts, to cause an action potential to take place. So we have to reach that negative 55 millivolts at this trigger zone to make that take place. So our graded potentials have to get this region of the membrane up to negative 55, and then an action potential will be sent. Okay, you can see this stimulus here, when we reach the trigger zone, we are below that threshold, so we would not have an action potential. Okay, so the size and amplitude we see up here is directly proportional to the stimulus strength. If we have a larger stimulus, the amplitude over here might be much larger. Okay, and what is causing these graded potentials? Okay, we have chemically gated channels, all right, and mechanically gated channels that may cause these action potentials. Gated channels, remember, if we have a sodium channel, gated channel, and we have, say we have a neurotransmitter, a chemical, that opens up a gated channel. And let's say that channel is sodium. Which way is sodium going to rush? Is sodium high or low outside? Yes, sodium is high outside. So sodium would rush into the cell. And what is sodium bringing with it? It is bringing a positive charge. So that would cause depolarization. Right? So these chemically gated channel that lets sodium in, if it was the opposite, if it let potassium, if this chemically gated channel allowed potassium, potassium is high inside and low outside, so potassium would rush out. So we would have a hyperpolarization, so an inhibitory signal. Okay, so that's what's taking place here to cause these graded potentials. We're getting depolarization, okay, more positive effect on the membrane potential, or hyperpolarization, inhibitory effect on the membrane. Okay? But if we're able to get enough excitatory change, enough depolarization to cause this region to get to negative 55 millivolts, what we call threshold, then we can send, the neuron will send an action potential. Okay, so let's look a little more on how we get up to this negative 55 millivolts. Looking a little bit, this shows you kind of that decrease in graded potentials, the strength. Okay, you can see here's the stimulus. It happens here, but as the signal travels throughout the cell body, what happens to the signal? It decreases, and this is happening because there is current leaking out and there's also resistance from the cytoplasm. So as these graded potentials travel through the cell, they are decreasing in amplitude. Okay. This is just showing the same thing. As we move through the cell, there is current leak, all right, and there is resistance from the cytoplasm, and that brings the amplitude of the graded potential down. All right, so let's get us up to triggering action potentials. And this was from that previous side. If we were measuring voltage where the stimulus is coming in to this neuron here, we can see, are we above -55? Are we above threshold? Because to cause the neuron to send the action potential, that signal down the axon, that electrical signal down the axon, we have to be above threshold here. And you see, as it travels through cell body, we're pretty much right at threshold, but as the signal comes through and gets to the trigger zone, the spot where we need to be at threshold, that -55, at threshold, we are below that threshold range now. Okay, and there is no action potential triggered. But look at this larger stimulus. We get a larger stimulus, okay, much higher and wider on the amplitude of this. Right? As it travels through, it does lose steam. It's a graded potential, but because that stimulus was greater, when we reach the trigger zone, we measure, we can see we now are above threshold, and that is going to trigger an action potential. Okay, that's going to be the trigger. Okay. No matter what we reach, if we reach -40, it's going to trigger. If we reach just -55, we trigger an action potential. Okay, it's just whether we're above that value or not. It's like a light switch. It's not a dimmer. Okay, you either turn the action potential on or it is not going at all. So let's take a look at this zone, this trigger zone, and where the action potentials are going to be triggered. Okay, if we reach threshold, what we're going to find here is a high concentration of voltage-gated sodium channels. Okay, and say this is a voltage-gated sodium channel. When the membrane potential is below -55, these channels are closed. There's a little sodium. Right? These channels are closed, but once we reach that -55 millivolts, that threshold value, that is when those sodium channels will open up. Okay, and what's sodium going to do? If there's a ton of channels here, there is a lot of channels. So a lot of ways for sodium to get in. If those channels open, then there will be a big rush of sodium in the cell right here at the axon HELOC, at the trigger zone. Okay, so that's what's happening. This trigger zone is kind of an integration center because it's basically, we've looked at the graded potentials and realized that graded potentials, we've seen one neuron. But you can imagine this neuron could be talked to. It could be getting stimulus from many, almost thousands of neurons here. Could be synapsing with this neuron. So there are many excitatory signals, many inhibitory signals coming at the same time. But if they all add up to -55 here, when we get to the trigger zone, we are going to open up those voltage-gated sodium channels. And remember, there are a high concentration, so there are lots of pathways for that sodium to come rushing into the cell here. Okay. So we already talked a little bit about depolarizing and hyperpolarizing from these graded potentials, but let's put a name to them. So EPSP, postsynaptic potential. So if you remember, the neuron comes in, and say this is a dendrite of another neuron talking here. I know this is a great picture, but say this is a neuron here, and this is the neuron coming into synapse. Okay, there is a synapse. So the postsynaptic would be what's taking place at this neuron. Okay, and so we have excitatory postsynaptic potentials. That would bring, excitatory would bring depolarization, like you see here. Okay. Inhibitory postsynaptic potential. If this neuron was sending an inhibitory signal, you would get something like that. And remember, there are many neurons talking to this other neuron. And so we can have many of these, but what's going to happen at that trigger zone is these are all going to get added up at the trigger zone. But remember, they also decrease depending on where they're taking place. And if they all add up to negative 55 millivolts, then that will open up those voltage gated channels at the trigger zone. Okay, and so how do all these values, we can get spatial summation of graded potentials. This is where we have 3 different neurons coming in, talking to this neuron, and they're all excitatory. But if we get a signal basically from all 3 of these almost exactly at the same time, simultaneously, as they travel through the cell, they're all traveling, and eventually they will meet up at the trigger zone, and these can be summed. All right, this one might be below threshold, this one might be below threshold, and this one might be below threshold if they were signaling at the same time. But because they're signaling at the same time, the addition, the summation of all of these may lead up to us to reach negative 55 millivolts at the trigger zone. Okay, so that's spatial over space. There are multiple neurons. This signal is not happening at the same space. Okay. Whereas temporal, we have the same neuron. The same neuron can fire almost simultaneously. Boom, boom, followed 2 graded potentials, stimuluses taking place almost right after each other. Here we see stimulus happens. All right, this is stimulus X1 and X2 coming from the same neuron, but we have a delay between the second action potential. So these 2 will not get summed. But if the action potentials happen almost simultaneously here, boom, boom, very quickly, when we reach the action or trigger zone, they may be close enough in time, temporal, to be summed together. Okay, so they're not coming from a different space, like spatial. That was coming from 3 different neurons, but almost the same time. This is happening where the same space, but happening at such a fast pace that they can be added up temporal wise. Okay, so this is what's happening at the cell body and the dendrites. We're getting all these excitatory and inhibitory signals. Okay? And ultimately, it's to determine whether there is enough signal at the right time to cause that action potential, that negative 55 millivolts, that threshold. Okay, and because what's this the cue for? This is the cue to open up those voltage gated sodium channels here at the trigger zone. Once we reach threshold here, all those voltage gated sodium channels open up, and if all those channels open up, we're going to go well above threshold. Okay, so we're going to result in an action potential once we reach threshold. And if we look at an action potential traveling down this long axon, if we were to measure the electrical value, remember action potentials do not decrease. So when I measure here, the value here, I haven't gotten, my amplitude's not down here. Okay, it's still up maximally. Okay, and this is what happens with an action potential. It basically is going to get re-upped. The signal is going to get re-upped all the way along the axon, and so we don't lose signal along the way. This is important because when we get to the end, if we kept on losing signal down this long axon, by the time we got to the end of the axon, if we lost signal, there would be nothing left. So this signal does not decrease as it travels down the axon. And we're going to see, because we get a re-upping of the signal all the way along the way of the axon. Okay, let's just look at this real quick. Okay, because the ions we're going to be dealing with are sodium and potassium. Okay. And what is going to be triggering these events are voltage gated ion channels. But let's see which one, if we can predict which ones are going to be in play here. Okay. If we have our little cell, our sodium is high outside, and potassium is high inside. Okay, which one's going to cause a depolarization, and which one is going to cause a repolarization or hyperpolarization? Okay, we have a voltage gated sodium channel, and we have a voltage gated potassium channel. Okay, so looking, if I opened up a lot of potassium channels, which way would potassium go? It is high inside and low outside, so potassium would leave. What does potassium bring with it? Okay, it brings with it its positive charge. If I'm losing positive, I'm going more negative. So as you can see, which of these is going in more negative, the depolarization or the repolarization? Yeah, so we can already associate this part of our action potential, this change in membrane potential, probably going to be devoted to our potassium channels. The opposite end, our sodium is high outside and low inside. When my voltage gated sodium channel open up, what's going to happen? Sodium's going to rush in, and what is it bringing with it? It's positive charge. If I'm adding positive to this negative environment, I'm going to make it less negative, which you see here with the depolarization. Right? So we can already associate what's going to happen. What ions are going to be involved with these different parts of the action potential. Sodium here, potassium over on this side. Okay? So hopefully we already got this idea that sodium channels would cause depolarization, where potassium channels would cause repolarization or even hyperpolarization. Okay, so now we got to add all the timing values and get the channels in here to look at to see how this action potential operates and continues down the axon.